The main type of cell that makes up the nervous system is called a neuron. These look very different from the cells we are familiar with thus far, in that there is an incredible amount of branching within each neuron, where cytoplasmic extensions project in many directions, allowing a neuron to respond to stimuli and transmit electrical impulses over very long distances. These are the very same signals that stimulate the muscle contraction we learned about earlier in the series. Neurons are amitotic, meaning they do not divide, and they have the potential to live 100 years or more, so the neurons we are born with last a lifetime. Beyond neurons, there are also supporting cells in the nervous system called neuroglia, or glial cells, which are not neurons, but rather wrap around delicate parts of neurons for protection. Let's get a closer look at a neuron now. First, the central part is called the cell body. This contains a nucleus and a nucleolus, surrounded by all the typical organelles we learned about in the biology series. Projecting from the cell body, we can see many dendrites, which produce a tremendous surface area for receiving signals. We also see a single axon. This initiates from an axon hillock and extends for some distance. For many neurons, the axon accounts for the majority of the length of the cell, sometimes extending macroscopic distances, at which point we can call them nerve fibers. At the terminus of the axon, there will be many terminal branches, sometimes thousands, which are the axon terminals that interface with other neurons, or with muscles to form the neuromuscular junctions we discussed earlier in the series. The axon is the conducting region of the neuron, in that it generates electrical signals for communicating with other neurons, starting at the axon hillock and traveling through to the axon terminals, also called the secretory region. When the impulse gets here, neurotransmitters are released, which will then either excite or inhibit nearby nerve cells. Many axons are protected by a myelin sheath, which also increases the speed at which impulses are transmitted, and nerve fibers covered in this manner are called myelinated fibers. Particularly in the peripheral nervous system, the myelin sheath is often made of individual units called Schwann cells. These first envelop the nerve fiber and then wrap themselves around it many times, which results in numerous layers of plasma membrane to protect the nerve. These are membranes which lack most of the channel proteins found in other types of cells, making them great electrical insulators and allowing them to increase the speed of electrical conduction down an axon. There are tiny gaps in between each Schwann cell where a bit of the fiber is exposed, and these are called nodes of Ranvier, and sometimes axon collaterals branch out at these gaps. Now, we want to be aware that there are several types of neurons, and just like some other anatomical features, we can classify them either by structure or by function. Going by structure first, neurons can be unipolar, bipolar, or multipolar. These terms refer to the number of processes extending from the cell body. A unipolar neuron has a single axon that quickly divides into proximal and distal branches, so this still technically qualifies as a single axon. One of these is a central process that goes towards the central nervous system, while the other is a peripheral process that acts as a sensory receptor. Then there are bipolar neurons, which have two processes, one axon and one dendrite, extending from opposite sides of the cell body. These are not very common, but they are found in certain places like specific parts of the eye and nose. Then multipolar neurons have three or more processes, precisely one of which is an axon, and the rest of which are dendrites. Around 99% of our neurons are of this type. Then we can describe function. A sensory or afferent neuron transmits information from sensory receptors towards the central nervous system. These are typically unipolar. Motor or efferent neurons transmit information from the central nervous system to muscles and glands. These are always multipolar. Then there are interneurons, which sit between the other two types and help shuttle signals around the system. These are typically multipolar neurons found entirely within the central nervous system, and most of our neurons are of this type. For any neuron, regardless of type, we can describe some basic common features. Any neuron will have a receptive region where a stimulus is received, typically at dendrites. Then there is a trigger zone, which initiates a conducting region where the electrical signal travels. This leads to a secretory region with axon terminals that release neurotransmitters. That covers the basics regarding the structure of a nerve cell. But how exactly does it generate electrical impulses? This could be discussed for hours upon hours, but let's go over a very basic summary here. First, we must understand the concept of membrane potential. For a more rigorous discussion of electric potential from a physics standpoint, check out my tutorial on that subject now. Otherwise, for our purposes here, we just need to know that opposite charges attract. So if there exist opposite charges near one another, work must be done to separate them. If these regions are already separated within a cell, there is an opportunity to use the potential energy that exists by virtue of their separation. This charge separation exists within nerve cells due to the concentrations of specific ions that sit inside and outside of the plasma membrane. So we say there is a potential difference across that membrane, and the membrane itself resists the current flow, as formally charged ions have difficulty traversing the nonpolar section of the membrane. However, there are ion channels in the membrane which can allow specific ions through at specific times. Some of these are non-gated, meaning they remain open. Others are gated, meaning they are closed and only open as the result of a particular signal. Chemically gated channels open when a specific neurotransmitter binds. Voltage-gated channels respond to changes in the membrane potential. And mechanically gated channels open when the receptor becomes physically deformed. When these channels open, ions can freely diffuse through, obeying the electrochemical gradient, which seeks to balance charge, so an electrical current is generated. A resting neuron has a resting membrane potential, based on the sodium and potassium ion concentrations in and out of the cell, due to their differing abilities to diffuse across the membrane, and it is maintained by sodium-potassium pumps that keep the concentration gradient as it is. But when a signal is received, whether from sensory input or a neurotransmitter from another neuron, there will be a change in this potential which can produce a signal. This signal will be either a graded potential, which operates over short distances, or an action potential, which operates over long distances, like the length of an axon. In the latter case, depolarization must exceed a particular threshold, meaning sodium channels must open and a sufficient number of sodium ions must diffuse into the cell. But if achieved, a nerve impulse will result, and a current will propagate along the axon towards the axon terminals. Repolarization will then occur where voltage-gated potassium channels allow potassium ions to exit the cell, which leads to a brief hyperpolarization, and then everything resets to original levels and positions. As for the impulse, when it reaches the axon terminals and neurotransmitters are released, these enter the synaptic space. A synapse is a junction between neurons or between a neuron and an effector cell. This is where communication happens. These synapses can be axodendritic, axosomatic, or axoaxonal, depending on where the axon terminals from the presynaptic neuron connect to the postsynaptic neuron. So that covers the basics regarding the action potential. We could get much more detailed and quantitative than this brief description, and perhaps we will dig a little deeper later in the series. But as we are currently focused on the big picture, let's be satisfied with this rudimentary understanding for the moment. Now that we know a bit about the structure and function of a neuron, let's discuss how these are assembled to form the nervous system. This system receives sensory input through receptors, integrates this information to decide what to do, and then generates a motor output, which is a response to stimulus. This is a lot more complicated than it sounds, so the nervous system has many components. First, it is divided into two main parts, the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and spinal cord, while the peripheral is all the rest, mainly the nerves that extend all around the body, which are bundles of axons. That means the central nervous system is the control center, where all the sensory information is integrated, after which a motor output is determined and implemented. The peripheral nervous system is what allows for communication throughout the body. The peripheral nervous system is also divided into two parts. There is the sensory or afferent division, which sends signals from receptors to the central nervous system, and the motor or efferent division, which sends signals from the central nervous system to all the muscles and glands to do its bidding. In turn, the motor division is also divided into two parts. There is the somatic nervous system, which sends signals to the skeletal muscles we consciously control, and the autonomic nervous system, which sends signals to smooth muscle and cardiac muscle that operates without conscious control. And finally, the autonomic nervous system is also split into two parts. Those are the sympathetic division and parasympathetic division, which operate in conjunction and typically have opposing functions. There is so much to discuss at each tier of structure that we have just mentioned, so let's go through these tiers one at a time, starting with the central nervous system.